I'm very pleased to introduce today's speaker, Andy Moeller, who, as I said, is a new postdoc in my lab. He comes from Yale University, where he was a graduate student with Howard Ochman working on gut microbiota, and that's what he's going to continue working on here. And he's very interested in interacting with everybody in the museum and thinking about different vertebrate taxa. So the great advantage of having new postdocs speak when they first arrive is everybody gets to know who they are and where their office is, and hopefully this promotes interaction. Andy did his undergraduate degree at the University of South Carolina. And as a graduate student, he had a NSF a graduate research fellowship, and published a number of great papers, presumably the contents of which he'll touch on, or at least some of them today. So we're very excited to start the first MVZ lunch with Andy, and the title of his talk is right up there.
Thanks a lot. Thanks for the introduction, and it's very exciting to be here giving the first MVZ seminar, so thanks to everyone for coming. The title of my talk is The Evolution of Human Integrated Gut Microbiomes. So this might be a bit of a departure from maybe more of the vertebrate-oriented talks, because most of what I'm going to be talking about today is bacteria, but hopefully it will be interesting. So when vertebrates first transitioned from water onto land, they were met with a whole new set of environmental challenges that they had to adapt to. And one of these adaptations was the evolution of the large intestine, which helps terrestrial vertebrates retain water from the diet. So if you look at the gastrointestinal tracts of fish, fish lack a true large intestine. And in the most extreme cases, the intestine is mostly an undifferentiated tube. But if you look at terrestrial vertebrates, tetrapods, there's an immense diversity of gastrointestinal tract morphologies with various different gut compartments that have evolved in response to different dietary strategies and different needs for the different organisms. So this new gut compartment represented a new niche for microbial colonization. And I want to emphasize the point that it's not just any niche for microbial colonization. It's arguably the best niche for bacteria that's ever existed in the history of life on Earth. So bacteria that grow in the gut, they grow to densities that we don't see anywhere else in the biosphere. So each human has about 100 trillion bacteria living on and inside them. And most of those are in the large intestine. And so it's not surprising that this extremely dense community of bacteria has become integrated with all different aspects of host biology. And this is an ongoing field of research, and it seems like every week there's a new connection between bacteria and host biology. So just to give a little history of this, in 2002 this paper came out that showed, in using germ-free mice as a model system, that mice that lack their bacterial communities don't really develop blood vessels in the large intestine. So angiogenesis is halted. And they actually found the bacterium that's involved in angiogenesis, or at least one of them, this bacteria called Bacteroidaceae, that simply by treating germ-free mouse with this bacterium, you can promote, during development, angiogenesis in the large intestine. And since then, there's been lots of work looking at the interactions between the gut microbiome and training the immune system. So here, another species within this genus, Bacteroidaceae, excretes a compound called polysaccharide A that helps maintain the homeostasis in the immune system. And obviously, bacteria also help in producing short-chain fatty acids from undigestible carbohydrates. So if you're a herbivore, you're able to be a herbivore because of your gut bacteria. So these observations of these relationships between gut bacteria and the host and the interactions imply a long history of coevolution between bacteria and hosts. And so what I'm interested in is exploring that coevolutionary history. So I'm going to talk about 3 different projects today. This first, the first of which was part of my PhD dissertation, and these second 2 are sort of ongoing or early stage projects, so hopefully you'll bear with me on that. So in part 1, I'm going to examine how the gut community has changed during human evolution. So I'm using humans as a starting point to start looking at the evolution of the gut microbiome, because we know much more about humans than any other group at this point due to the Human Microbiome Project. And so in part one, I'm going to be considering the gut microbiome as sort of like as if it were a morphological character, and doing traditional comparative approaches to infer the changes in the composition of the gut microbiome that occurred as humans diversified from other species of apes. In part two, I'm going to look at the possibility that gut bacteria and hosts have actually co-speciated with one another. And in part three, I'm going to talk a little bit about how we think bacteria are being transmitted across generations and maintained over evolutionary time. So the basis for the part one is that African apes house clues about the ancestral state of the human gut microbiome. I probably don't need to emphasize this too much to this audience because everyone's familiar with comparative approaches. So here's the phylogeny of humans and the African apes. And the idea is if we can look in populations of gorillas, chimpanzees, bonobos, and humans, for instance, we can perhaps identify consistent differences in the composition of the gut communities of these different groups, and then using the phylogenetic relatedness of the host, infer the changes, the changes in the composition of the gut community that occurred as the host diversified. So to answer this question in part one, we first have to answer a couple of descriptive questions. And those are what lineages populate the gut microbiomes of African apes, so simply what bacteria are present in African apes, which wasn't known before we did this work. And also, not just what's there, but what are the relative abundances of these different taxa within their hosts. So what's there and how many of each are there relatively. And so to accomplish this, we sampled a broad range of African ape populations throughout equatorial Africa. And so we're focusing on the African apes, so we don't have orangutans here, unfortunately. So we have gorillas sampled in Cameroon, for instance. We're going to focus on a chimpanzee population from Tanzania, and then a bonobo population in the Democratic Republic of the Congo. And the approach that we're taking to reconstruct the microbial communities living in these hosts is a next generation sequencing approach, which starts with a microbiome sample. In this case, it would just be a fecal sample that's collected in the field from these hosts. And we take the fecal sample into the lab, we extract DNA using a bead beating procedure, and amplify the 16S RNA gene present in the sample. So 16S ribosomal RNA is a universal gene in all bacteria. Every bacterium has a copy, at least one copy of this gene. And each bacterium has a slightly different copy, so it's a great phylogenetic marker that we can use to describe the diversity present in these samples. So after performing these PCR reactions, we sequence the libraries on the Illumina MiSeq or HiSeq platforms. And from there, we can do some bioinformatics, mapping our reads back onto the reference databases in order to identify all the species and all the taxonomic groups that are present in the sample, and also get a sense of the relative abundances of these different bacterial groups within the sample. So at the end of the day, the data looks something like this. These are two hypothetical samples. I've just completely made this up. But just to give you an idea of what the data might look like. So here, each column would be a sample. So these are two samples from two different hosts. And each color represents a different bacterial taxon. And so you can see, for instance, in this host, this group would be more relatively abundant than in this host. And so with that in mind, I just want to show you basically all the data from my PhD in one slide. And so these are all the gorilla samples that we sequenced. And each line, they're pretty skinny here, it represents a different host. And each color in this plot represents a different bacterial order. And we can make these plots at different taxonomic levels, but for this slide I chose order. So you can see there's a lot of a lot of diversity in gorillas. These are the bonobo samples that we have, and chimpanzee samples. And then we combined or compared our data with a human microbiome data set that was produced by another group. It was produced by Jeff Gordon's group in Washington University in St. Louis, who's really a leader in the human microbiome field. And so his group had sampled 3 human populations. Human population living very remotely in the Amazon rainforest in Venezuela. Human population living in Malawi, in rural Malawi. And also a big population of humans living in urban areas in the United States. So if you kind of unfocus your eyes and stare at this for a while, you can see pretty clearly that there are some sharp distinctions between the different host species. So gorillas have this technicolor pattern of diversity, whereas if you look at humans from the United States, there's just this big smear. And also, if you look more closely, you can see that there's even distinctions among humans in the United States versus humans in Malawi and Venezuela. So to visualize this in a different way, we can use principal coordinates analysis. So here is a principal coordinates plot based on the relative abundances of bacterial genera recovered from each sample. And I want to note that these 2 axes only explain about 1/4 of the variation in the data set. But even with that in mind, you can see some clear distinctions. Humans here, and bonobos and chimps kind of clumping together here, and the gorillas mostly down here. And if you do statistical tests on this, it's very clear that each host species is harboring a compositionally distinct gut microbiome. But also, I wanted to point out that if you look at these upside down blue triangles here, these are humans from the United States. They seem to be forming a distinct cluster, even relative to other humans, those sampled from Malawi or Venezuela. And so it looks like something weird is going on in humans from the United States, where we're more divergent from apes than we would expect, and also more divergent from other human populations in terms of our gut constituents. So another way to look at that would be to plot microbiome dissimilarity. Here it's just Bray-Curtis dissimilarity of genus level abundances. If there are any ecologists in the room, you might know about that. As a function of evolutionary divergence between the hosts. So here I'm plotting a line that's only based off of wild living apes, so I've excluded humans from this analysis. So if you look within species, gut communities tend to be relatively similar. Bonobo versus chimp, a little more dissimilar still, and if you look at pan, either bonobos or chimpanzees versus gorillas, they're quite dissimilar. And so we can ask, where do humans fall on this line? And they fall sort of close to the line, but if you look at humans from the United States, they're always significantly above this regression line. So it looks like that humans from the United States their gut communities have diverged from the ancestral state more so than we would expect based on evolutionary divergence of the hosts alone. Next I'm going to show you which taxa are actually maybe driving this pattern, at least in terms of which taxa differentiate humans from wild apes. And so this is, again, treating the gut microbiome as if it were a morphological character. We're looking at the relative abundance of each genus in the gut of all of these different host species, and performing statistical tests. We're looking for bacterial taxa that are overrepresented in one host group relative to all the other host groups. And so, for instance, everything here above this branch represents bacterial taxa that are overrepresented in human populations relative to all the wild ape populations, each wild ape population or species individually. And for this analysis, I'm only showing the bacterial taxa that differentiate each human population from the wild apes. So these aren't taxa that are specific to humans from the United States. These are taxa that are either over or underrepresented in humans in all human populations relative to wild apes. So we found many changes in the abundance of bacterial taxa occurring along the human lineage. And I just want to point out a couple that are maybe more interesting. We found over a 5-fold increase in the relative abundance of Bacteroides in humans. And other work has shown that Bacteroides is associated with hosts eating diets rich in animal fat and protein. We also found a corresponding decrease in the relative abundances of Methanobrevibacter and also Fibrobacter, both of which are associated with the fermentation of plants. Methanobrevibacter is not a bacterium, actually, it's an archaean, it's a methanogen. But it removes the end product of fermentation, thereby speeding up fermentation. So it looks like the human gut microbiome has become more specialized for a meat-based diet. And this, again, I just want to reiterate that this is not only humans from the United States, but this is true of all the human populations that we surveyed. So I want to go back to the idea that humans from the United States are harboring gut communities that are highly divergent from those of other human populations, and to ask why are human gut microbiomes in the United States so divergent from those of wild-living ape species? There are hypotheses out there, one of which has been publicized in this book recently by Martin Glaser at NYU, who claims that the overuse of antibiotics is fueling our modern plagues. If there's not more sensational title than that, I don't know what would be. But this book and these ideas, there's a lot of this now, especially in the popular press, about antibiotic use is causing asthma, autoimmune diseases, maybe even autism. But it's based on not a whole lot of evidence, actually. So we wanted to look, at least in the gut, to ask if we can find examples of these missing microbes. So how do we know if humans are actually missing microbes relative to wild apes? And again, I think that phylogenetic comparisons can resolve this question pretty clearly. So if we saw this pattern where humans have very low levels of diversity and all the wild ape species have high levels of diversity, it would be a very clear inference that there was some decrease in diversity along the human lineage. And that's exactly what we see when we look at the gut microbiomes. So this is the mean relative number of bacterial genera recovered per individual. And in wild apes, you can see that the mean's about 85 bacterial genera per individual. But in humans from the United States, we're only recovering about 55 bacterial genera per individual. And what surprised me about this is we didn't just see a decrease in humans from the United States relative to wild apes, but also humans living in Venezuela and Malawi. And presumably these people in Venezuela have very limited exposure to antibiotics or to modern medical practices. So I take this as evidence to suggest that there's been a reduction in diversity in the human gut microbiome even before the advent of modern medicine and antibiotics. I think it likely predates the divergence of extant human populations, but there's been probably a secondary depletion. This depletion's been exacerbated by the use of antibiotics in modern medicine. And I want to say too that this reduction in ancestral diversity in the human microbiome wasn't just evident at the genus level. We can look across all taxonomic scales. So if we look at phylum, class, order, family, genus, and 97% OTU, which is the bacterial word for species, we can see this reduction holds across all these taxonomic levels. And more so that humans from the United States always have the lowest levels of diversity of any human population or any wild ape population. So with that, I just want to summarize. Part one really quickly. The main take home of this is that the human microbiome has undergone a transformation since the divergence of humans and wild apes. We harbor higher frequencies of bacterial taxa associated with meat eating, and lower frequencies of taxa associated with plant digestion. And of all the African apes, humans contain the lowest levels of microbial diversity. And humans in the United States contain even lower levels of microbial diversity. Recently people have looked too at other westernized populations. So this is also true of people living in Europe. So urban lifestyles really are what I think is causing this secondary reduction that we observed in the humans from the United States. So it's nothing specific to humans from the United States necessarily. And I just wanted to mention too that the differences we observed among the microbiomes of the different host species appear to be robust to variation in lifestyle, as well as geography and disease status. So we've also looked at sympatric and allopatric populations of chimpanzees and gorillas and found that they maintain their distinct gut communities even when living in sympatry. And we've also looked at chimps who are infected with SIV, an immunodeficiency virus related to HIV of humans, and found that although they do experience some alterations in their gut communities, they still maintain the chimpanzee typical gut community. Okay, so in part one, I've argued that the gut microbiome, the community composition of the gut microbiome, recapitulates host phylogeny. But I want to say that that pattern alone doesn't necessarily mean that bacteria and hosts have co-evolved. So I think there's a lot of confusion in the literature about this, that using this word coevolution when really the evidence for coevolution isn't quite there. So it could be that more closely related host species are simply selecting more similar sets of bacteria from their environments. And the bacteria themselves don't have to be evolving in response to the host at all. They might just be hanging out in the environment and selected every time a new host is born. So in part two, I want to start to look at the possibility that that bacteria are evolving in response to their host's evolution, to try to get at this question of coevolution between hosts and bacteria. And so one way to look for this is to test for patterns of co-speciation between bacteria and their hosts. And so probably maybe the most famous example of co-speciation between a bacterium and an animal is the aphid Buchnera system. So Nancy Moran and Paul Baumann showed in the 90s that the phylogeny of aphids and their endosymbiotic bacterium mirror each other perfectly, suggesting that there was an origin of this endosymbiosis a long time ago, hundreds of millions of years ago. As aphids have diversified, the bacteria have diversified alongside the aphids as well. So, excuse me, I should probably say just as background, aphids inside them harbor these specialized organs called bacteriocytes, in which live Buchnera, which helps aphids generate essential amino acids from their sap-based diet, which is pretty much deficient in all the things you need to survive. And so it's a pretty remarkable system. And so this is extremely strong evidence that the bacteria are evolving in response to their host speciation, right? It's hard to think of another explanation for this pattern of co-speciation. So I would really love to see if we can test for this pattern in gut bacteria in humans and in apes. But there was a problem initially that the 16S amplicon sequencing that we were using doesn't provide the resolution that you need to test for co-speciation between humans and apes and their gut bacteria. And this is because 16S is just too slow. It's evolving too slowly to test for co-speciation. So there have been attempts to calibrate the bacterial molecular clock using aphids and other insects that have endosymbiotic bacteria. And estimates from these methods have put the rate of evolution of 16S somewhere around 1% sequence divergence per 50 million years. So it's extremely slow, and if you think, gorillas and humans maybe only 18 million years, that's only a few nucleotide substitutions are going to differentiate the 16S sequences recovered from gorillas and humans. And humans and chimps, they might have 100% identical 16S sequences. So there's really no way to test for co-speciation using 16S. So in order to get at the question, we have to use a different approach. And so this is work that I started during my PhD, and alongside, so this method was developed alongside a postdoc in the lab named Alejandro Quintero, and we're just working on this now, so this is in progress work. But so to get at this question of co-speciation, we're going to use a GyrB amplicon sequencing. So GyrB is a protein coding gene, and it's much more variable than the highly conserved 16S. And so GyrB is a protein in this gyrase complex that is involved in relieving tension put on the DNA strand by the helicase during DNA replication. So this is another universal protein. All bacteria have this protein, but because it's a protein coding gene, it's going to be more variable than 16S. So we're going to use, we developed these family-specific bacterial primers to amplify GyrB sequences from 3 different bacterial families. And these were chosen due to their abundance in the gut, and also they're known to be important in a lot of the interactions between host and bacteria. So the 3 families that we chose are Bacteroidaceae, Bifidobacteroidaceae, and Lachnospiraceae. And this GyrB amplicon sequencing, it works just like 16S amplicon sequencing. You just cross out 16S and put GyrB in its place. And at the end of the day, instead of getting a sense of the relative abundances of all the different bacterial taxa that exist in the gut, we're going to have a complete catalog of all of the strains within a single bacterial family, within each of these bacterial families. So we're looking at a much finer resolution at the diversity than previously. So I want to point out that a single 16S sequence might have over 10 or more GyrB strains within an identical sequence. And so we sequenced a bunch of samples from gorillas, humans, bonobos, and chimpanzees, and got a lot of data back. And this is just a short segment of one of the alignments. This is for the Bacteroidaceae, looking at the GyrB sequences. We've recovered about 760 abundant strains from the 4 host species. And you can see, if you can see this, it's maybe a little small, but these asterisks here represent conserved sites, and there's a lot of white space here, so there's a huge amount of variation in this protein coding gene that we can use to infer the phylogenetic history of the bacteria. And the question we want to ask is, does the phylogenetic history of the bacteria mirror the phylogenetic history of the hosts? And so for the Bacteroidaceae, it appears the answer is yes. So we observed a large clade of GyrB sequences from gorillas that seem to be sister to all other GyrB sequences, represented by this collapse clade. And within this collapse clade, we found 3 main clades of GyrB strains. And so I'm going to show you those 3 in succession here. This is the first of those 3. And I should say, too, the relationships among these 3 clades within this collapse clade were not well resolved by our analyses, so we're still unsure about that. But yeah, within this first clade, you can see that humans, bonobos and chimpanzees are sister to one another, with humans as that group, as you would expect under a co-speciation scenario. And then the other 2 clades, human sequences appear to be absent. But again, bonobos and chimpanzees are sister to one another. And the final clade, again, bonobos and chimpanzees are sister to one another, with gorilla sequences as the outgroup, and humans are missing. So maybe this is a way that we can start to identify specific strains that are missing from the human population. Also, I wanted to point out too that this pattern is sort of analogous. You could think of it like a gene duplication, where bonobos and chimpanzees seem to have 2 closely related types within this one clade that have maybe perhaps co-speciated in parallel. And so in the case of the Bacteroidaceae, we seem to have some good evidence that there has been some co-speciation between hosts and their bacteria. And we can also look into the Bifidobacteroidaceae. And we see a similar pattern where the phylogeny of the strains more or less matches the phylogeny of the host, with the exception of this one clade in which bonobo and gorilla sequences from the Bifidobacteriaceae are sister to one another. So this potentially represents a transfer from bonobos into gorillas. And of all the populations that we sampled, the bonobo and gorilla populations are the only two that share a border. They share a border on the Congo River. And it's possible that they were in sympatry at some point in the past. And so I think this may represent a transfer from bonobos to gorillas. And also want to point out, you can see a huge diversity of strains within each host species, so which is consistent with sort of radiation of strains after host speciation events. Finally, we looked at the lacnosporation, and we found absolutely no signal of co-diversification whatsoever. So this tree, each branch is colored by the host species it came from, as in previous slides, and you can see everything's all mixed up with each other. And so it looks like the lacnosporaceae are being frequently transferred among host species, as opposed to, most extremely, the Bacteroidaceae, and maybe less so the Bifidobacteriaceae. And this is somewhat anecdotal, but I was trying to think of reasons why this differs so much from the other two groups. And lacnosporaceae are spore-forming bacteria, whereas Bifido and Bacteroidaceae are not spore-forming. So some bacteria can form these hard spores that allow them to survive harsh conditions, such as might be faced when bacteria is moving from one host species to the other. So it's possible. I'd like to look at more bacterial families in the future to see if that pattern holds up, but it's possible that the life histories of the bacteria are influencing the propensity with which they're able to be transferred or co-speciate. So just to summarize part two, there are a range of evolutionary histories within the gut microbiome. There are some gut bacterial lineages, like the Bacteroidaceae and Bifidobacteriaceae, that appear to be co-speciating with their hosts, whereas others are frequently moving among host species. So I don't think there's one type of gut bacterium, there are many types. And something that excites me about this is that by utilizing the known divergent states of the host species, we can get much better estimates of calibrating the bacterial molecular clock, which is something that's been almost impossible to do before because of the lack of a bacterial fossil record. And the question that I'm interested in going forward is this co-speciation phenomenon a common feature of vertebrate diversification, or is it some idiosyncrasy of apes? So I'm really interested in looking at other groups to see if we can look at coevolution between bacteria and vertebrates in a broader context. So just very briefly, in part three, I wanted to start talking about how these bacteria are being transmitted across evolutionary time. So if it's true that bacteria are co-speciating with their host, it implies some mechanisms to ensure the fidelity of the transmission of bacteria across generations. And so I'm going to consider two alternative scenarios. One is strict vertical inheritance from mother to offspring. And this is the strategy that aphid Buchnera uses. So Buchnera is transmitted vertically to the mother. It's also the strategy that our own mitochondrial endosymbionts use. It's strict vertical transmission from the mother. The alternative might be social transmission, where socially interacting hosts are exchanging bacteria all the time, and there's no vertical transmission along maternal lines, but vertical transmission in the sense that a population is maintaining a bacterial symbiont as a whole. And so to differentiate between these two, we're going to use the well-studied field system at Gombe Stream National Park in Tanzania, famous for Jane Goodall's work. It's been a field site for over 50 years now. And there's several populations, social groups of chimpanzees living in Gombe, and the genealogies of these chimpanzee families are very well understood, so it allows us the opportunity to look for vertical versus horizontal transmission. And I just want to say too, this work was done in collaboration with Ann Pusey and Stephen Forrester at Duke, and also Michael Wilson at the University of Minnesota. And this is something that's under review right now, so it's not published yet, but hopefully will be soon. And so for this project, we sequenced bacterial DNA from 160 fecal samples that were collected over 10 years from chimps whose genealogies were known, so we know the relatedness among the chimps. And we asked a very simple question, which is, do chimpanzees share more bacteria with their maternal line than they do with unrelated hosts with which they socially interact? And the answer is no. So chimpanzees appear to share as many bacteria with completely unrelated hosts of their own social group as they do with their own maternal line. And I was sort of amazed when I saw this result. You couldn't get closer to identical. So on the y-axis here is, again, Bray-Curtis dissimilarity, looking at the presence-absence of species, and we have within and between maternal lines. So this is suggesting that vertical transmission isn't the dominant mode of inheritance, but just to give a little bit more evidence for that, we were able to look at the social environment of the chimpanzee population when the fecal samples were collected. And so on the x-axis here, we have a measure of chimpanzee sociability, which is just the mean half-weight association index. It's essentially the average amount of time, percentage of time, any two randomly chosen individuals in the social group spent together. And so when chimps were more social, their microbiomes were less dissimilar. So they were more similar. They harbored more similar gut communities during periods of high sociality. And also their gut microbial communities tended to be more diverse during periods of high sociality. So just to sum up these last few ideas, I think although this is something that obviously needs to be looked at more closely and in more host groups, I think our working hypothesis now is that gut microbial constituents are primarily horizontally acquired from conspecifics. And even though the initial inoculum might come from the mother during birth, that the microbiome housed by an adult tends to be largely acquired horizontally from conspecifics. And I want to point out that the exchange of bacteria within host populations through social interactions, but not between host populations, can also lead to the pattern of co-speciation, just like strict vertical inheritance might. However, this horizontal transmission decouples bacterial fitness from host fitness. So bacteria and hosts have independent fitness interests in a scenario in which bacteria are being horizontally transmitted. So this might slow the rate at which co-adaptation occurs. I'm also interested in the possibility that host sociality may be an essential mechanism by which diverse microbial flora maintain over time. Because social transmission should eliminate any sort of bottlenecks that might exist when bacteria are being strictly vertically inherited. Sort of like meta-community theory, in which you have many, many patches all connected to one another as opposed to just a few patches. So just a final summary slide of the three main parts of my talk. The community composition of the gut microbiome recapitulates the host phylogeny. Some of the bacteria in the gut appear to have co-speciated with their hosts, whereas others seem to be transferred all the time horizontally. And I think the primary mode of inheritance, at least in chimpanzees, of gut constituents is horizontal through social interaction. And I just wanted to briefly mention what I'm planning to do at the museum. And the first part of the work I'm planning to do is to extend some of the work I just showed you to look at a broader range of vertebrate groups, to not only examine how the community composition of the microbiome is transformed during vertebrate evolution, but also to examine this, the possibility that co-speciation between bacteria and vertebrates is a commonality of vertebrate life. And then the second project that I'm interested in is to get a little more experimental and to actually test for signatures of co-adaptation. So everything I've shown you, I haven't actually proven co-adaptation in any sense between bacteria and hosts. All the data are consistent with it, but in order to really measure co-adaptation, it's going to require experiments. So I'm interested in using germ-free mice, which have been developed now and it's a pretty straightforward technique now. So to take bacterial communities from different species of wild mice within the genus Mus, of varying divergences from Musculus, and to, through fecal transplants, inoculate germ-free lab mice with these microbiota and measure fitness of both the host and bacteria under different circumstances. So with that, I just want to acknowledge my advisor, Howard Ockman, who has been extremely good advisor to me. Also my committee, Nancy Moran, Paul Turner, and Andrew Goodman. Alejandro Quintero is the postdoc who developed the GyrB sequencing. That was his, he spent an entire year troubleshooting that, and this work would have not been, it's going to be useful, I think, for a lot of different projects, including this co-speciation stuff. And Kim, Eli, and Megan, people in the lab that trained me and helped me, Ockman and Moran Labs in general. Then these collaborators who I mentioned previously, and also now the Michael Nachman and the Nachman Lab for having me. So, thank you.
I'll let you, yeah.
I have a question about your basic fecal sampling procedures. It seems like it's possible that you might be getting a biased sample if you're interested in the total gut, by going to the end product, so to speak.
Right.
And I wonder if anybody has checked by sampling the actual gut from different positions
Right.
of the gut and comparing it with this fecal sample to see if there is a bias.
Right, so people have done that experiment. Actually, Taichi in Michael's lab has a paper soon to be published looking at mice. People have looked at different gut compartments and to determine which gut compartment is the fecal representative of, if any. And they found that the feces tends to be representative of the large intestine, which sort of makes sense. But so I consider all of this work really looking at the microbial community within the large intestine. So we're not looking at other gut compartments here. So that's a good point there. So something to consider. Oh, yeah.
Kind of going along with that. Well, actually, before I ask that, is a fecal transplant as uncomfortable as it sounds?
Probably for the so the way it's done now is through gavaging.
Gavaging? Remind me never to sign up for that one.
Yeah. So for a human, I think they're working on just having a pill. But for mice, it's done with gavaging largely. And so it's extremely unpleasant, I'm sure. Yeah.
Right. Well, actually, my question was,
Okay.
how does your gut, how do these fecal samples say, how do they change from day to day, depending on what you're eating, and then seasonally during the year?
You mean the gut community host, or if like?
Yeah, like when you sample your,
Yeah.
or gorilla number 15.
Right.
In that, if just within one individual, like how much does that gut flora vary
So there
day to day and year to year?
There are big fluctuations in terms of the relative abundances of taxonomic groups at higher scales. So like if you look at the relative abundances of phyla, you can see vast fluctuations within an individual, but I think the strain level community membership is fairly stable.
So diet doesn't affect your gut.
Oh, diet definitely affects your gut microbiota quite a bit. So if you shift from eating a meat-based diet to a plant-based diet, you'll see corresponding shifts within the same day, actually. And people have looked in humans. But even with that variation, samples collected from the same individuals over time are almost always more similar to one another than samples collected from different individuals. So there does seem to be an individual signature that it definitely has.
I have two kind of quick questions. The first one is, which gorillas did you say you sampled?
So here I talked about western lowland gorillas, but we also in some other projects sampled eastern, the mountain gorillas living.
Okay, that's what I was wondering, because their diets are pretty different.
Yeah.
And then my other question was, For the chimpanzees that you sampled, are any of them part of the populations that are known to like eat a lot of red colobus monkeys, for example? Because I wonder if their microbiomes might be a little more similar to humans eating meat or other animals that are eating a lot of meat.
So these chimpanzees in Gombe do hunt
Right.
and eat monkeys. So I'm looking at this now where we have dietary information for all of these chimps, and so I'm trying to see if I can look at the effect. We know when they've gone on a hunt and eaten meat, and we know which individuals ate the meat, and so we're looking at that now, but I don't really have a clear answer yet for that.
Talk again when you do.
Yeah, we can. Yeah.
So your, the data on social transmission, that was from 16S?
Yes, those looking at identical, 100% identical 16S sequences.
So if you were going to use a faster evolving gene like the gyrase, for example,
That would be ideal.
do you think you would see differences between those different groups and the degree of vertical transmission versus horizontal through social transmission?
Yeah, I think that will be the real test for distinguishing vertical versus horizontal. So I hope I didn't overstate it here, but yeah, looking at 16S because it's such a coarse marker, it's really hard to get at that. So we have some samples that we sequence the gyrase, for which we sequence gyrase B, and so I'm going to look at that. That's ongoing still. But, yeah.
Is there any relationship at all between diet diversity in the host and their gut microform diversity?
So people have looked in different systems at this. I think there was Dan Bolnick had a paper recently looking in stickleback at dietary diversity and gut microbial diversity, and actually found a inverse correlation, if I'm remembering that correctly, which was unexpected. And if we look in chimps, we have this measure of dietary evenness. I didn't show these data, but dietary evenness appears to be positively correlated with microbial diversity, which, so it seems like different host groups, even within a host species, different sexes might have different, diet might have a different effect on levels of diversity. So it's sort of up there, yeah.
So you emphasize the fact that the different species tended to be consistently different from each other. But if I remember your first PC plot, there were a bunch of gorillas up in the bonobos and chimpanzees, right?
Right. So, yeah, the PC plots are slightly misleading, I think, because you're reducing the variation. So if we've also made like these 3D PCA plots, and they actually, you see the gorillas pull out.
But it also suggests that some gorillas are still more closely related in their gut microbiota to chimpanzees and bonobos.
Definitely, definitely. So gorillas seem to be, each gorilla is one of two sort of community types. One of which looks more like the traditional ape or the standard ape gut microbiome. And then the other has a lot of environmentally derived bacterial taxa, which is common with a lot of herbivores. So yeah, I think gorillas are by far, within gorillas, there's an extreme amount of variation. Within bonobos and within chimpanzees, they form very tight clusters, but gorillas are very variable. So,
Geographically related?
so even within gorillas, all from the same field site, there's a tremendous amount of variation. Yeah.
So do you think there's any possibility that the beginning of the use of fire and cooking and so on has reduced human diversity? And if so, has anybody looked at like people who have these raw food diets?
I get emails about this all the time. Well, not all the time, but several. People in the paleo diet want to know is there.
No, not paleo,
But,
raw food.
Yeah, raw food. So I think it's possible that there was a transition in the gut microbiome corresponding to the advent of fire and cooking. The human gut, if you just look at the anatomy, is pretty different even from chimpanzees. We have a reduced large intestine, a larger small intestine, which is typical of like a carnivore, more carnivore. But I think it's an open question. I mean, how much of this is really evolution of the host versus just our diet? So, experiments are good. Yeah.
So is there a mechanism by which humans, or any other host for that matter, can maintain the preferential microbes and delete those that are
Absolutely,
not
yeah.
preferential? And
This is an extremely active area of research. So basically the host immune system can recognize certain bacteria as good, or that they should be there, and as others as bad, and that they shouldn't be there, and either upregulate or downregulate inflammation along the epithelium of the gut in response to that. And how that is working has been only just recently starting to be worked out. There's a paper maybe like 3 or 4 months ago in Science from Andy Goodman's group that found a single phosphate group in a part of a protein along the cell membrane of Bacteroidaceae. If you remove that phosphate group or leave it on, that changes whether or not the host will recognize that the bacterium is good or bad, and either upregulate or downregulate inflammation. And so that's just one example, but presumably there are other mechanisms like that where the host is recognizing certain features of these bacteria. But yeah, it's largely unknown, I think, how it's actually happening.
It certainly seems like another possibility for coevolution, because you could imagine that a non-beneficial microbe would be tapping into this as well, and there would be constant switches between the marker.
Yeah, it's sort of like a red queen thing.
Yeah.
In the spore-forming bacteria that didn't show the co-speciation pattern, there were still clades that were mostly of one species. Can you use that information to get at the frequency of
That's a good
horizontal transmission?
Yeah, so something I'm working on right now is to try to get a good molecular clock to this GyrB sequence. And since I'm at Berkeley, I'm going to use a lot of Bayesian approaches to this. But yeah, we could look at the branch lengths within a host species and estimate kind of a minimum amount of time that was needed since a transfer event for that diversity to arise. Yeah, it's a good idea.
Most of your analyses seem to be based on relatively few taxa in any one round of analysis, like 5 or 6 genera or whatever. I mean, that's a very small percentage of the total number of taxa that are actually present in the gut. Is that true? I mean, is there any way you can expand that and take a look at rare species that might be doing important things?
So with the first part of the talk, with the 16S analyses, that does give a very broad look at all the bacterial diversity that's there. But for the second part where I looked at the co-speciation, that, you're right, we only focus on 3 families, and as of right now, can't think of a good way to do that in a really high throughput way. You kind of have to design primers that amplify specifically different bacterial families. The holy grail of this, I guess, would be if you found some universal gene that had a perfectly conserved region flanking extremely variable region. Then you could maybe do that, but so far, yeah.
Are you at all concerned about the gyrase being targeted by certain kinds of antibiotics? Like the fluoroquinolone antibiotics?
In terms of like what we might find?
Yeah,
or
that there might be evolutionary shifts. If you start using that gene, say in humans or something, you might find some really weird
You mean as a
results.
molecular clock as a problem?
Yeah.
That's an interesting point. Actually, I haven't thought of that.
Yeah.
So,
I don't know if it's that particular.
yeah, we should look maybe to see this the motif that, or this, the region we're amplifying of GyrB to see if it's maybe a target of potential selection in response to antibiotic use. That's a good point.
Okay, well, if you have more questions, you know where to find Andy. He's in the office next door. If you have your favorite fecal sample from a vertebrate, you can drop it off. I'm sure he'll be happy to take all your crap. Thanks again, Andy.